we have so lucky to have an expert about viruses and pandemics on our campus. And Dr. Laura Newcomb received her PhD in 2001 from the University of Wisconsin-Madison, studying RNA expression and cell cycle regulation. Always maintaining a keen interest in viruses, in 2002, Dr. Newcomb was awarded an NIH Ruth L. Kirstein National Research Service Award postdoctoral fellowship to explore the physiology of an important viral pathogen, influenza virus, with the aim to identify new ways to combat these pathogens. Dr. Newcomb, lucky us, joined the Department of Biology at CSUSB in 2007 and has continued her funded research on influenza virus involving CSUSB students along the way. Thank you. We are so grateful, Laura, that you took the time and you are going to share your expertise and experience right now. So what I'm going to do, I'm going to stop sharing my screen and I'll turn over to Laura.
Hide that. Okay, let's see. Okay, so hopefully everybody can see my screen. So thank you so much for inviting me to give a presentation to everyone today. And as you know, we're currently undergoing a pandemic due to the 2019 coronavirus, and pandemics are when we see spread of disease over a large region. I'm going to talk today not only about our current situation, but I'll talk a little bit about probably one of the most notorious pandemics, the 1918 influenza pandemic, because influenza is the virus we study in my lab. And then I'll end with just a little bit about the future and how we can try to better prepare ourselves. And so we have been here before. This is a clipping from 1918 in Illinois, where they did take these non-pharmaceutical interventions like closing down schools and churches and theaters as we saw increase in flu victims. And over here we have a picture of some makeshift hospitals that were set up to take that overflow. And so as we talk about non-pharmaceutical interventions, I just want to show a graph that maybe some of you have seen that is an example from two different cities in 1918. And so the solid line is showing you Philadelphia, where they experienced their first case in September 17th, and they didn't implement these non-pharmaceutical interventions like sheltering in place and physical distancing until 16 days after their first case, even having a parade in between that time. And so you can see, this is the death rate per 100,000, that there was a huge peak which would overwhelm the healthcare system. Now, in contrast, St. Louis, maybe they had a little bit of warning because they didn't see their first case until October 3rd. And so they rapidly implemented these non-pharmaceutical interventions, and you can see it really did help spread the slowing of the infection so that the healthcare system wasn't overwhelmed. And so I'm a molecular virologist, so I do have to tell you a little bit about the molecular biology of the virus that's responsible for the 1918 influenza pandemic. And obviously that's the influenza A virus. And this virus is a negative sense RNA genome virus. And what that means importantly is that the enzyme that replicates the influenza genome is an RNA-dependent RNA polymerase, and it has no proofreading ability. What that means is that during replication, it makes a lot of errors, and so you have a lot of changes in the genome of influenza, and that's termed genetic drift. Influenza is also a segmented genome, so the genome is in pieces. There are 8 different segments that get packaged into the virus. And so if one host cell is infected with two different influenza viruses, there can be a reassortment of those segments, and that can result in new viruses emerging, and that's termed genetic shift. I just want to point out this complex that packages the genome, that's the viral RNA-dependent RNA polymerase, and the nucleoprotein. This complex is the VRMP. It's responsible for the viral gene expression and replication, and is the complex of study in my lab. I also want to point out two other viral segments that encode two important viral surface proteins, and these are the hemagglutinin and neuraminidase. And so these proteins are important to bind the host cell receptor and enter the cell, and to exit the host cell. These proteins are also how the influenza virus is categorized into subtypes. And so you may have heard reference to things like H1N1 or H3N2. That's referring to what type of hemagglutinin and neuraminidase proteins are on the surface of that virus. Also, these are the proteins that your immune system will produce neutralizing antibodies against, and are the ones that are targeted by those yearly vaccinations. In addition to these molecular mechanisms to provide the ability of the virus to change, another contributing factor is that influenza infects many different animal hosts. It does not just infect us. So I have listed here just some of the different animals that influenza can infect. Now, the natural reservoir for influenza is thought to be wild aquatic birds, but pigs are susceptible to infection with avian influenzas. And obviously the pig is not a bird. So as the virus evolved to replicate more efficiently inside the pig, it's thought that's how human influenzas emerged. However, the pig remains susceptible to infection with human influenzas, swine influenzas, and avian influenzas, and is considered by many to be a mixing vessel for those reassortment events. And that's actually what we saw in 2009, where we did have a pandemic of a novel H1N1 influenza. That virus had segments of avian, swine, and human influenza origin. Now, the natural reservoir can also infect our poultry friends. We have a lot more contact with our domesticated birds than we do with wild birds. And so although rare, direct avian to human transmission has occurred. Notably, in the recent past, some viruses of concern were the H5N1 highly pathogenic avian influenza, which was first characterized in 2003 with just a few cases, but you can see a very high mortality rate. 2015 was the year the world saw the most cases of that. Again, a very high mortality rate, but fortunately, that virus only infected people who had very, very close contact with those infected domestic avians. Now, by 2019, the world saw just one case, so hopefully that's not going to cause any problems. But of course, in the meantime, an additional avian virus has infected some humans, causing high pathogenicity. And so now researchers are also focused on looking at an H7N1 highly pathogenic avian influenza. So these molecular mechanisms that I talked about, the antigenic drift, which is when the polymerase, while replicating, makes some errors, and this can cause changes to the surface proteins, and these changes might be changes that our immune system has not seen. In addition, the antigenic shift is when you have, it is very sensitive, when you have two different viruses that infect the same host cell. So you can think an avian influenza and a human influenza infecting a swine cell, and then reassortment can happen and you see emergence of a new virus with a different combination of properties that maybe our immune system does not recognize. And so indeed, both of these mechanisms of change have occurred to cause viral pandemics. So the 1918 influenza pandemic was a virus that was of complete avian origin, whereas the 1957 and 1968 were reassortment events. The 2009 novel H1N1 pandemic was considered a triple reassortment because the virus had human, avian, and actually two different swine origin influenza viruses. And so influenza was responsible for numerous pandemics, the most notorious being 1918, but is still, of course, a concern for an emerging pandemic. And I hope that the brief bit of molecular biology I've told you about allows you to understand why it's actually quite easy for influenza to gain new molecular characteristics that could result in new things for our immune system and a possible pandemic. So here we are again with schools closed and giving a talk to apparently a lot of people, but it's through Zoom, so it's hard to tell. And, you know, we've had some cities with hospitals overwhelmed. We're closing campuses, but this time the culprit's not influenza. This time the culprit is different. It's a coronavirus, specifically SARS coronavirus 2. Again, this is an RNA genome virus, but it's a little different from influenza. It's considered a positive sense, not really important. But what is important is that coronaviruses are a bit unusual. They have very large RNA genomes, about over twice the size of the influenza genome. So the RNA-dependent RNA polymerase of coronavirus is actually a bit unusual in that it's coupled with a subunit that does have proofreading activity. So it does not make as many errors during replication as the influenza virus does. But of course, given that it's a virus and it replicates very rapidly, producing lots of offspring, there still are mistakes that are made even with proofreading. And so the virus still does undergo genetic drift. The virus also is not segmented. There's just one genome segment. So unlike influenza, there's no reassortment. But that doesn't mean that there's not recombination between different coronaviruses. There's a mechanism I'll tell you about more on the next slides called polymerase switching, which happens quite frequently with coronaviruses because they have such long genomes. And so if a cell is infected with two different coronaviruses, it's very easy for those genomes to recombine and produce new viruses from genetic shift. Coronavirus also has a nucleoprotein. I just say that because my lab is interested in those proteins in viruses. And then the last protein I want to talk to you about is the spike protein, or the S protein on the surface of the virion. That's the protein that's responsible for binding the host cell receptor and entering the cell. And that would be the protein that our immune system should develop neutralizing antibodies against, and what we would be targeting with a vaccine. And so similar to influenza virus, another thing that makes coronaviruses good for causing pandemics is that they also infect many different animals. So that gives them a lot of opportunity for those molecular mechanisms of gaining new characteristics to occur. In particular, reservoirs appear to be the bat and maybe also the rodent, although some feel the rodent obtained the virus from bats. But there is a lot of diversity of coronaviruses residing in bats. And a lot of the diversity comes in that spike protein that's responsible for targeting the cell. So for a long time, it was known that coronaviruses could jump to human, but they always caused mild infection. So wasn't much of a concern until 2003 when we saw the SARS outbreak. And if you remember that, that was contained in part by shutting down Toronto for a while. This coronavirus had very high mortality rate, but fortunately was contained. We saw a different coronavirus jump in 2012, and this was primarily in Saudi Arabia. This is the Middle East Respiratory Syndrome, MERS coronavirus. Again, very high mortality rate, very severe symptoms, but fortunately was contained. Now, when the paper that I took this figure from came out, this was actually in early March before knowledge of the current coronavirus pandemic. And these authors were actually quite worried by a different coronavirus that jumped from bats into pigs and was actually causing severe disease in pigs. And so this was a concern that it may jump to humans. Now, of course, that's not what happened. What happened, not yet anyway. What has happened currently is that a different coronavirus in bats has jumped to human, possibly through an intermediate, maybe the pangolin, although this has not been conclusively confirmed. But what we do know is that the virus now spreads very easily among humans. And so the same molecular mechanisms that I told you about in influenza are also at play with making coronaviruses so variable. You have the antigenic shift where the polymerase enzyme makes some mistakes, and that can result in changes into the protein, into a new protein, a new virus that our immune system hasn't seen. Now again, coronavirus doesn't do this as often as influenza, but it still is very much a driving force in coronavirus evolution. Antigenic shift is when a single cell is infected with two different coronaviruses. So I have them indicated by the gold or silver genomes. And because these genomes are very, very large, the polymerase enzyme, which is indicated by this little purple circle here, it cannot replicate the entire genome at one time. It falls off and has to reanneal and continue replication. And so if there are two different coronaviruses in that same cell, it can bind to the other coronavirus template and finish replication, resulting in these recombinant viruses that now have very different characteristics than the original virus. And this could result in a new virus that our immune system is not accustomed to. And so I do want to show you some of the changes that are appearing in the S protein, that's that spike protein that's important for binding the host cell and entering the cell, that are found in the SARS coronavirus 2. And so what you're looking at here is the genome sequence. In pink is the sequence that encodes the spike protein. They've put the amino acid sequence here. And what I want you to look at is a close-up of the receptor binding domain. And here they're indicating one-letter amino acid codes, color coding all the same amino acids for you. So what's highlighted are the changes in this receptor binding domain. And so these are different than what's found in SARS, but some of these changes are found in the original bat SARS related coronaviruses, but they also Some are present in pangolin coronaviruses, which is where some think perhaps that might have served as an intermediate. It doesn't necessarily matter exactly where they evolved from. There's multiple mechanisms for changes in sequences, but what is important is that these changes have given the S protein the ability to bind the receptor much better. Because it can bind the receptor better, that means it can infect the cells better. Now it can target host cells that don't have necessarily a high abundance of this receptor. So it has opened up the host cells for the virus. Another change is that there's an insertion here of a polybasic cleavage site, which allows the virus to use a very common host protease for entry. And this protease is actually very common in our upper respiratory tract and explains some of the reasons why this new coronavirus 2 is so highly transmissible. And because it is so highly transmissible, I just want to put a plug and shout out for non-pharmaceutical interventions, because they really do help spread. They really do help stop the spread of the virus. And so this is just looking at the effect of masks. And so these researchers looked at countries that had a masking culture in the dark green and countries with no masking culture in the gray. The graph here is showing you day 1 is considered the day that that country had 50 confirmed cases. And you can see there's a clear trend with countries that wear masks have a slower daily growth rate compared to countries that do not wear masks. It's a very simple non-pharmaceutical intervention, and it does work. And so as these authors conclude, and they have other analysis as well, universal masking is urgent in the COVID-19 pandemic. And so, you know, my kind of statement lately has been zoonosis happens. We live in a world with animals, we consume animals, we have animals as pets, we breathe the same air as animals. Zoonosis happens and will continue to happen. And I've only talked to you about influenza and coronavirus as 2 examples, but there's many others like Ebola virus, Nipah virus, hantavirus that you know, should also be of concern for jumping. And so in my lab, we focus on influenza virus. So this quarter, and apparently through the summer and next, some of my students will be switching to more online projects and are looking more into influenza zoonosis. And so this is a picture showing all, well, not all, but many of the animals that are infected by influenza. And one thing I want to point out is the jumping of influenza from humans into our domestic animals, including canines. And although it hasn't happened yet, there are a number of researchers that are very concerned about canine influenza jumping back to humans. And so, you know, zoonosis happens. Humans live near animals all around the globe. And just because we had the emergence of a pandemic from one part of the globe now doesn't mean that our culture might not cause the emergence of a pandemic in the future. And I, for example, could be patient number 1 for that canine influenza jump. So how do we prepare for this inevitability? In my opinion, we need to invest much more money in surveillance to monitor both the human population and the animal reservoirs for presence of these viruses. We need to focus on treatments. I've highlighted that because that's an area of research in my lab. We're focused on identifying antiviral targets and looking at new antivirals, and treatments help give time to generate vaccines. So right now there is no coronavirus vaccine, but hopefully we can get one to have antibodies that will neutralize coronavirus. Now there are, however, vaccines for influenza. we have the influenza yearly vaccines. There's actually 2 different formulations. Most people get the inactivated virus, which is a dead virus that comes in the form of a shot. But if you have sensitivity to or allergies to eggs, then you may get the live attenuated virus, which is through a nasal spray. And so that does replicate a bit inside the person to produce that response. And actually that vaccine was taken off for a few years while they had to reformulate to make sure your body produced enough neutralizing antibodies. Now, the vaccines are engineered so that you produce antibodies against 3 to 4 circulating strains of influenza. Again, they target those HANA surface proteins that can change very rapidly. So vaccines aren't very helpful against an emerging subtype of influenza, particularly because vaccine production time takes quite a while, although there's a lot of work and effort to speed up vaccine production time. But as you're seeing currently, vaccines are not really ideal for an emerging pandemic. So for an emerging pandemic, antiviral therapies are usually more effective because they will inhibit a viral protein activity, and they tend to be effective against even emerging subtypes. And so they can be your first line of defense while a protective vaccine is made. So for influenza, this is just a cartoon depiction of the influenza life cycle. And for influenza virus, the first class of antivirals targeted the influenza M2 protein, which is an ion channel that is required for this fusion event to release the genome. Now, this class of antivirals is completely ineffective now. 99% of circulating influenza viruses are resistant to that class of antivirals. The second class of antivirals for influenza you might be more familiar with. The brand names for those are Relenza and Tamiflu, and those both target the neuraminidase activity of influenza and inhibit that budding. And so while those are still effective, it's really just a matter of time before we see resistance emerging. And that's because even as early as 2014, it was already seen that there were genome segments that could encode resistance not only to the M2 ion channel inhibitors, but also to the neuraminidase inhibitors. And so I had a great master's student who wrote this review article on promoting targeting the viral nucleoprotein as an antiviral target. And, we weren't the only laboratory promoting this complex as being a good antiviral target. And indeed, in 2018, a new antiviral was approved for influenza. You may have seen the commercials. It's called Xofluza, and it targets that VRMP complex. So my lab studies the activities and interactions of the influenza viral ribonucleoprotein complex to look for targets for inhibiting the virus. In particular, we focus on the nucleoprotein, which this is a crystal structure of a monomer of the nucleoprotein with some domains that are important for NP function and could be targeted with antivirals. And so, for example, these residues that are highlighted are important for the oligomerization activity of NP that's required to form this complex. The RNA binding domain is important to bind the RNA genome, which is essential. And for my lab, we're very interested in this body domain of NP, which has been implicated in binding the small molecule inhibitors nucleosin. But in my lab, we've also found that those same amino acids are important in NP-PB2 interactions. And so we hope to look for additional inhibitors of this interaction to inhibit the activity of this complex. I'm also very interested in studying the coronavirus replication machinery. And so this is a depiction of the coronavirus RNA-dependent RNA polymerase. You may have read about how an antiviral called remdesivir is doing, showing some promise against coronavirus, and it actually targets this complex. So interestingly, I was already starting to look into coronavirus prior to our current pandemic because I really love cheetahs. And in the 1980s, feline coronavirus was actually killing a number of cheetahs at zoos. And over this past December, I was fortunate to get to go to the Cheetah Conservation Fund in Namibia, which was founded by Dr. Laurie Marker, the author of this book I'm showing. And we talked we talked while I was there about feline coronavirus because it's currently causing problems for cheetahs in Somaliland, where Dr. Marker is trying to set up another cheetah conservation site in addition to her site in Namibia. So I just want to plug another activity that's happening during Research Week this week, and there's another webinar on Thursday at 9 o'clock in the morning. The West Coast director from CCF will be giving a talk on cheetah and humans sharing a landscape. And so hopefully I'll eventually get to at least maybe feline coronavirus as a model. And so lastly, I just want to thank you all for listening and thank my lab and all my current and former students and past and current funding. And so with that, if we have time, I can try to answer any questions. Thank you.
So if you have a question, you can either type it to the chat or raise your hand. And we'll try to see if anyone, I don't even know how to manage this. But.
I see the chat.
So there's a question. Do you see the question?
I do see the question. So the first question I can't.
Could you read the question?
Oh, so the first question, sorry, is currently clinical testing is underway on mRNA-based vaccines. Whoa, it's going too fast. How likely are these to be effective in producing a protective immune response in humans? And can you speak to how this works? So I'm not, I'm more of a molecular virologist than a vaccine researcher, but my understanding of these mRNA vaccines is that they will be, the mRNA will be translated into the viral protein. And so from there, it's very similar to how the current vaccines work that may put in proteins already. It may be more efficient because maybe you'll get more of the protein and produce a more robust response. I think that's at least the hope. Okay, so second question, do we know the mortality or morbidity for coronavirus? So I do have one slide. Again, I'm not an epidemiologist either. Let's see if I can get my other slide. Here we go. But I do have a quote from a preeminent epidemiologist who's the director for the Center for Infectious Disease Research and Policy at the University of Minnesota, who gives some simple math about, there's 320 million people in the United States. Even if the infection rate is just 50%, which I think I've given you some indication that with the characteristics of the S protein, it's probably a little bit better than that, but that would be 160 million people. And let's just say the mortality rate is only 0.5, that still is 800,000 deaths in the next 6 to 18 months. And so, this is an interview that he had with a local paper. So, see if there's any other questions.
Yeah, I can read it. It seems that data are clearly available showing that masks work. Why did the CDC initially recommend we not wear them?
So the reason, at least my understanding, the reason the CDC initially recommended not to wear masks was primarily to avoid not having healthcare workers having masks. So it wasn't so much that, you know, masks don't work or aren't needed. It was more that masks are really needed for the first responders, and you know, we can't take the masks away from the first responders. So that's my understanding of why they said that. And then another question is, how long do you think it will take to develop a vaccine that works? Again, I'm not really a vaccine researcher, but you know, it will take time to find a vaccine. Another question, I know it's an immunological question, but based on, whoa, too fast. Based on past experience with the flu, how likely serum immunity is effective? Okay. Okay. Yeah, so then there's some questions about vaccines and actually getting immunity. Is this going to end up being a virus that can change frequently? So will we need yearly vaccines? It might. I think it's really too early to tell. And someone asked a question about the survivability of the coronavirus. And again, I think, you know, we need to wait to see the data on that. But the data that's been coming does show that it can survive for quite a while. But, you know, you do still need to be exposed to a number of the viruses to be infected.
The next question asks, is it possible for the virus to stick to clothing?
Again, I haven't done that kind of research and haven't looked into that kind of research so much on the survivability, but I believe the suggestion, if you're an essential worker, to definitely just shed everything, throw it in the wash, and hop in the shower. So until we know more, that might be the best thing to do.
Great. And no more questions, I suppose? No? Yes. One more question? Two more questions?
Well, yep. Upon recovering from the virus, how much immunity do you have? That's, again, a question that really is unknown at this time. So I'm not an immunologist, but not every antibody that you produce against a virus is always neutralizing, and not every antibody lasts all the time. So, you know, those coronaviruses that cause the common cold, we can typically be reinfected with those. So that question is still out. And then Lysol kills the virus, but definitely don't drink it. Soap works too. It's probably better to just wash your hands with soap. Okay.
Okay, so I think this is the last question, and clearly you sparked a lot of interest, and I'm going to go back to sharing my screen. Thank you so much, Laura. And I wanted to thank you, and I think you've earned a great round